Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we started discussing about cellular signaling in which we have covered various different signaling pathways that are very crucial for different cellular events which can be during the developmental stages or which can be during the adult stages. So first we discussed about the basic signaling pathway, the basic cascade that is followed by the cell so that it is able to communicate with some other cell or with itself, or it is able to convey whatever environmental changes are being done in its extracellular matrix on the intracellular side. So we learned about different cell surface receptor proteins such as the three main categories, the GPCRs, the RTKs, the enzyme coupled receptors, and the ionotropic receptors, which are ion channel coupled receptors. So all those three different receptor proteins, they use different mechanisms so that they can switch on the molecular switches, which are going to get activated, and then they are going to switch on and off certain genes, the target genes for that particular signaling cascade. So first we learned about GPCRs, the G protein coupled receptors. Then we learned about different signaling pathways which get started or activated when the receptor tyrosine kinases are activated. Then in the last class we learned about JAK-STAT signaling pathway as well as the TGF beta signaling pathway. So the JAK-STAT signaling pathway is mediated by cytokines. So cytokine receptors are the ones which get activated to activate the Janus kinases downstream, which will activate STAT proteins, which are the latent transcription regulators. And in the same way, TGF beta signaling also occurs. So in the TGF beta signaling, the receptors for TGF beta extracellular signal protein are the serine threonine kinases. And the serine threonine kinases, once they get activated, they are going to activate SMAD proteins downstream, and those SMAD proteins are going to do their downstream job. And those SMAD proteins, they also have their self inhibiting negative feedback loop, which will inhibit or which will disrupt the whole signaling pathway. So JAK-STAT signaling and TGF beta signaling, they resemble each other a lot. The signaling that we are going to discuss today is Notch delta signaling, which is a very important signaling pathway, and which is very distinct from the all of the signaling pathways that we have learned till this date. And it is the signaling pathway which is very straightforward and shortest known till now, so that the signal will be conveyed from cytoplasm to the nucleus. So the most straightforward pathway to convey a signal from the cytoplasm or from the extracellular environment to the nucleus, shortest route that can be followed is followed in the notch delta signaling. So the notch delta signaling pathway is the topic of focus today. Notch delta signaling. Both of them are receptor proteins present on the neighboring cells. Okay, so this notch delta signaling is very distinct because it is the contact dependent signaling. Means the two cells which are communicating with each other, those cells are, those cells are in the vicinity, means they are the neighboring cells, they are physically in the contact. So that is why this is contact dependent signaling. So for this signaling to occur, the both of the cells should be in the vicinity, both of the cells should contact each other physically. So this notch delta, both of them, when they interact with each other, those receptors, when they interact with each other, the cells basically they are coming in contact, and that is why this is known as contact dependent signaling, or it is the example of juxtacrine signaling. Right, when we learned about different in the first lecture, when we learned about different types of signaling, we learned about juxtacrine signaling. So this is the example of juxtacrine signaling. So this the general role of notch delta signaling is that it is useful in cell-fate choices. So when the cell has to commit to certain cell fate, when it is going to decide that it is going to be either neural cell or epidermal cell, notch delta signaling is very helpful, and it also regulates pattern formation during the developmental stages. During development, right? So the best known role for notch delta signaling is self-head choice, and this was first studied in Drosophila. So notch delta signaling, the best known role for it is in Drosophila. So neural cells in the Drosophila, they develop through this notch delta signaling. So today we are going to learn about development of neural cells in the Drosophila, how they use notch delta signaling pathway so that the cell fit will be decided. So in the Drosophila, a single sheet of the epidermal precursor cells exist. So let's assume that those cells are epidermal precursor cells. So a single cell out of this sheet of the epidermal cell decides that it is going to be the neural cell. So when this certain cell, let's assume that this cell is deciding that in future it is going to be a neural cell. When the certain cell decides to be a neural cell, so this is our future neural cell. Okay, so when it commits to a certain cell fate, it inhibits other cells, the neighboring cells, to not to do the same. So let's say that this particular cell has committed to being a neural cell in future. So it is going to signal the adjacent cell, the neighboring cell, that you are not going to commit to becoming a neural cell. And this inhibition of the neighboring cell by the future neural cell to not to become, to not to commit to becoming a neural cell is known as lateral inhibition. So this neural cell, the future neural cell, is going to signal all those neighboring cells that you shouldn't commit to being a neural cell. So that phenomena is known as lateral inhibition. Okay, so lateral inhibition is the inhibition by the future neural cell to inhibition of the inhibition of the neighboring cells to the future neural cells, that they shouldn't commit to becoming a neural cell in future. So the cell fate of them will be epidermal cells. So they shouldn't commit to being a neural cell. That is what we refer as lateral inhibition. So all of the rest of the cells, they are going to be epidermal cells. Okay, so those epidermal precursor cells, they will stay as it is, and only a single cell is going to be committed to becoming a neural cell. So this lateral inhibition, this lateral inhibition is done, and it is possible due to this neural cell which expresses a protein known as delta. So delta is a single pass transmembrane protein, the cell surface receptor. So it is expressed by this future neural cell, and due to the expression of delta over this neural cell, the other cells which are surrounding this neural cell, the delta expressing cell, so those other cells, all of them, they are going to start expressing notch receptor, right? So delta receptor, it will interact with notch receptor, and that is going to activate the protease, which is situated on the plasma membrane of the notch secreting cell, and that protease will cleave the intracellular domain of the notch receptor, and that is going to do the signaling. So this is how notch delta signaling is quite crucial for formation of neural cell. So let's now learn about in detail how the notch delta signaling works. So let's assume that this cell is a cell which is going to be neural cell in future. That is why it will express delta. So this is the delta protein, and let's assume that this particular cell is the cell which is not going to be a neural cell in future. So it is the epidermal cell, and that is why it is expressing notch receptor. So once this notch and delta, those receptors, they interact with each other. Well, I forgot to tell you, notch receptor is a heterodimer. Okay, so once those notch and delta receptors, they interact with each other, protease which is present on this plasma membrane of notch expressing cell, that protease is known as gamma secretase. This protease will cleave the intracellular cytoplasmic. So this will cleave the cytosolic tail. Okay, so this cleaving of cytosolic tail Okay, so let's assume that so this is the extracellular domain, this will be transmembrane domain, and this will be the cytoplasmic domain, right? So the gamma secretase will cleave the cytoplasmic tail and it will be released in the cytoplasm. And once it is released in the cytoplasm, it is going to translocate to nucleus and it will bind to a DNA binding protein. And after the binding to this DNA binding protein, it is going to convert that particular protein from a transcription repressor to a transcription activator. Okay, and that protein is RBPSUH protein. So this protein is actually a repressor protein. So this is a DNA binding protein and it is a repressor protein. But if to this protein the cytoplasmic tail of Notch receptor binds, then this repressor protein is converted into the activator protein. Okay. So the cleavage, okay, so cleavage has a very particular role in Notch receptor or in the Notch delta signaling. So this Notch receptor undergoes three cleavage events in its whole lifespan. Okay, so from the biosynthesis to the signaling part, it goes undergoes three cleavage phenomena. So, three cleavage phenomena from the synthesis to the signaling part. Okay, so this signaling, unlike the RTK signaling or the GPCR signaling, this signaling is irreversible because once this cytoplasmic tail is physically cleaved. So now Notch receptor cannot recover, right? So this once the cytoplasmic tail is cleaved, the Notch receptor, the part of the Notch receptor which is present extracellularly and this delta receptor, they will be internalized by delta expressing cells. So the Notch, as I told you, undergoes three cleavage events. So let's list them down, and those three cleavage events are very important. The very first cleavage event, that proteolytic cleavage that a Notch receptor undergoes, is during its biosynthesis. So when this receptor is being synthesized and it is being prepared in the Golgi apparatus, it gets cleaved in the Golgi apparatus and it makes a homodimer. Oh well, yeah, I told you that is a it is a heterodimer, right? So it forms a heterodimer. Let me just confirm it once whether it is a heterodimer or a homodimer. I'm getting confused. Um, just a second. Okay, yeah, I was right. It is a heterodimer. Yeah. So this is a heterodimer. Okay, I just got confused. Okay, so the very first cleavage that the Notch receptor undergoes is during the biosynthesis, and it gets converted into a heterodimer. This cleavage converts this Notch receptor into a heterodimer, and this cleavage is done in the Golgi apparatus during biosynthesis. And once this particular heterodimer of the Notch receptor is formed, now this whole receptor, this fully mature or fully formed receptor, which is also known as mature receptor. This mature receptor will be, it will be transported to the cell surface over the plasma membrane of the cell. And once it is transported to the plasma membrane, at some point of time, it will react with the delta receptor. And once it reacts with the delta receptor, the other two cleavages are going to get triggered. The cleavage of the extracellular domain of the Notch receptor, that will be done by some extracellular protease, and the cleavage of the cytoplasmic tail of the Notch receptor will be seen, which will release the cytoplasmic tail inside the cytoplasm to do its downstream job. So let's have a look at the schematic given in the Bruce buds so that this whole cleavage phenomena is quite clear to you all. So here you can see this is the Notch receptor being synthesized inside the cell. Okay, so this is the Golgi lumen. So this is the Golgi body, and this is the lumen, and this is the cytosolic part. So the whole cell is like this, and this is the Golgi. apparatus. Okay, so this notch receptor has a 36 EGF-like domains. So this 36 EGF-like domains, these are the ones of the notch receptor and delta receptor which interact with each other. Okay, so due to this domain, it is possible the interaction of delta and notch is possible. So this is now a single, it is a monomer, right? So here this red arrowhead, it indicates the proteolytic site. So a protease will come and it will cleave off this particular part. It will cleave at this particular residue. So once it cleaves this particular residue, now it is going to form a heterodimer. Here you can clearly see it is forming a heterodimer, and now this heterodimer, so this heterodimer is formed, means it is a mature protein. So from the Golgi lumen, it will be transported over the cell surface. Well, we haven't discussed the transport of the proteins yet, so we will be discussing that very soon in the cell biology series. So this protein will be transported over the cell surface, and now it is sitting there waiting to interact with notch protein, with the delta protein at right time. So when the delta protein comes, it interacts with this particular monomer, this EGF-like domain, 36 EGF-like domain, and the binding of this domain with the delta receptor will expose the second proteolytic site, which was there on the extracellular part. So see, here is the second proteolytic site, but it was hidden due to this particular domain. And once this domain binds to the delta receptor, now this particular site will be exposed, which will which will be cleaved by the some extracellular protease. So the extracellular part will be separated, and on the intracellular part, the cytoplasmic tail will be cleaved off, and this cytoplasmic tail is going to migrate to the nucleus, and it is going to do its downstream job. So there are three cleavage events. The first one is done during the synthesis of the protein, and due to first cleavage, maturation of protein is seen. So protein gets formed, and it forms a heterodimer. The second cleavage event is in the extracellular domain part, and third cleavage creates the cytoplasmic tail, which is going to do the signaling part. The intracellular signaling job will be done by this cytoplasmic tail. So this cytoplasmic tail will migrate to nucleus, and it will bind to the protein, as mentioned before, RBP-SUH protein. This protein is a DNA binding protein, and it was a repressor before, but due to forming a complex with the cytoplasmic tail of notch receptor, now this will turn into the activator, and this whole transcriptory activator complex is going to bind to the element in the DNA, which is the notch responsive regulatory sequence. And once it binds to this regulatory sequence, the target genes, the notch target genes, they will be transcribed. Okay, so this is how notch delta signaling is very different, and it is the shortest route, because unlike in other signaling, we are activating some G proteins, or we are activating some enzymes, or we are activating some kinases, right? So we are activating either of the molecular switches, the GTP binding proteins or the kinase cascades. So that those signaling cascades are longer, much longer, but the shorter and very straightforward way is provided by notch delta signaling. Imagine a receptor is residing on this plasma membrane. It interacts with the other cell surface receptor of the neighboring cell, and simply the cytoplasmic parts get cleaved, which is going to migrate into nucleus itself. So there is no involvement of other protein, other molecular switch, which is going to do the activation of transcription. The cytoplasmic tail, which is being cleaved itself, acts as the regulator, the regulatory. It is doing the regulatory job, right? So this is a very straightforward and shortest way of doing the signaling. As that is why notch delta signaling is very unique, and again it is unique because it is juxtacrine signaling. So the contact of the delta and notch receptor is very crucial for the signaling to happen. The other unique thing about the notch delta signaling is that usually that the signaling pathways that we learned till now were the pathways where some extracellular signal molecule is going to come and bind to the receptor, right? But in this case, the signaling is done by both cell surface receptor proteins. The delta protein is also a cell surface receptor, and notch is also a receptor. So this delta protein is acting as a signal for the notch, and it is going to bind this notch receptor and going to do the signaling. So this molecule, the extracellular signal, which is delta in this case, it is the transmembrane protein, means it is embedded in the plasma membrane, unlike the other extracellular signals such as growth factors, such as growth hormones or cytokines. They are released in the bloodstream, or they are released in the extracellular matrix, and then they will bind to the receptors of their on the which are present on that particular cell, right? So this is also a very unique aspect of the notch delta signaling. So with this, we are done with notch delta signaling, and this is very crucial because I will be mentioning that this signaling again in the when we will discuss about developmental biology. So this signaling is quite crucial for the in the Drosophila cells for the development of neural cells. So I'll be mentioning this again in the developmental biology series. So we are done with this signaling part. In next lecture, we'll learn about Wnt signaling, which is again a very important and unique way of signaling. So we are done here. Next class, we'll discuss about Wnt signaling. Till then, see you. Bye bye.